Hello, everyone, and welcome to today's ASBMB Breakthrough webinar, Surprising Enzymatic Role of OB fold Containing Proteins in Translational Fidelity. I'm Chenming Jiang from Publication Department at the American Society for Biochemistry and Molecular Biology, and I'm thrilled to be your moderator today. This webinar is part of our Breakthrough webinar series, sponsored by the ASBMB journals. Specifically, today's session is brought to you by the Journal of Biological Chemistry, JBC, one of ASBMB's esteemed journals, which has been publishing high-quality science that elucidates the molecular and the cellular basis of biological processes for over a century. Before we dive in, a quick reminder, you are welcome to submit questions at any time. Use the Q&A feature at the bottom of your screen. We'll address them during the dedicated Q&A session after the presentation. This webinar is being recorded, and the recording will be available on the ASBMB website. It's now my pleasure to introduce today's speaker, Dr. Karin Musier Forsyth. Dr. Karin Musier Forsyth obtained a PhD in chemistry from Cornell University. She was an American Cancer Society postdoc fellow in the laboratory of Paul Schirmer at the MIT and a faculty member at the University of Minnesota from 1992 to 2006. In 2007, she moved to Ohio State University, where she is the Ohio Eminent Scholar in Biological Micromolecular Structure and Professors of Chemistry and Biochemistry. She currently serves as Director of the OSU Center for RNA Biology. She has published over 200 research articles, book chapters, and reviews, and has served as an Associate Editor of JBC since 2018. Without further ado, please join me in welcoming Dr. Musier Forsyth. Dr. Musier Forsyth, the floor is yours.
Okay, great. Well, thank you, Chenming, for that nice introduction. And I'm really excited to be here today and to have this opportunity to tell you about this project that we're actually really excited about. And the main players in this work are highlighted here, Rylan Watkins, Hannah Lathrop, and Morgan Bauer. So just as a reminder, I want to just remind everybody that aminoacyl-tRNA synthetases are, housekeeping enzymes that charge tRNAs, and here shown here is the two-step reaction. So they activate a specific amino acid with ATP to form this enzyme-bound aminoacyl-adenylate. That amino acid is then transferred to the cognate tRNA, which then goes on to interact with the elongation factor in protein synthesis. But aminoacyl-tRNA synthetases are challenged by similar amino acids. So for example, some amino acids, and often misactivate amino acids. So some amino acids, such as isoleucine and valine, differ by only a methyl group. Others, such as valine and threonine, differ by only a hydroxyl group. And even amino acids which seem to have pretty unique structures, like proline, are really the same molecular volume as cysteine, and alanine is a smaller amino acid that is also misactivated by proline tRNA synthetase. And I'm highlighting these because this is the system that we're going to start out focusing on today, proline tRNA synthetase. And many synthetases are also challenged by non-proteinaceous amino acids, which are metabolites, and often very abundant metabolites in the cell. So as a result, the aminoacyl-tRNA synthetases can sometimes misactivate non-cognate amino acids, forming the incorrect aminoacyl-adenylate. If this happens, there's a chance for the synthetase to perform a function known as pre-transfer editing, so to basically to hydrolyze this incorrectly charged aminoacyl AMP at this step of the reaction. But in many cases, the synthetase will go on to mischarge the amino acid, so you'll get the incorrect amino acid on a particular tRNA. And then in this case, about half the enzymes have a proofreading activity that's called post-transfer editing. And this post-transfer editing can actually happen in two ways, cis and trans, and I'll explain what that is on the next slide. Next slides. So in the case of proline tRNA synthetase, as I mentioned, its cognate substrate, amino acid substrate is proline, but it can misactivate cysteine, which is the same molecular volume, and alanine, which is smaller, and so can fit into the enzyme, active site. And so the synthetic active site of what's shown here is E. coli proline tRNA synthetase, and the synthetic active site will, activate all three of these amino acids. E. coli proline tRNA synthetase then has this domain, a separate domain that we call the INS domain, because it's an insertion domain within the catalytic, active site of the enzyme. And it turns out that this insertion domain has the catalytic activity to hydrolyze alanine that's been mischarged onto tRNA pro. So it's an ALA tRNA pro deacylase domain. But this domain cannot hydrolyze cysteine, and instead cysteine is post-transfer edited by a homologous domain, so called Y-back. So that's when we discovered this activity, that's what this protein was annotated as in the database, and we've just maintained that name here. But so Y-back is a cysteine tRNA pro deacylase. And this is what I mean by cis editing. So CIS editing is when the editing domain is appended as part of the aminoacyl-tRNA synthetase, and trans editing can happen by freestanding editing domains, or sometimes by synthetases in trans as well. And so it turns out that, okay, this is the example of E. coli proline tRNA synthetase, which has a cis editing domain and one trans editing protein. But there are some bacteria, such as Colobacter crescentus, shown here, where the synthetase itself does not have the cis editing domain, and instead this organism encodes two trans editing proteins, one that hydrolyzes alanine off of tRNA pro, and we call that ProXP-ALA, and again the homologous Y-back protein, which deacylates cysteine that's been mischarged onto tRNA pro. So when we discovered this, and some of this early work in this area was published in JBC, what we found was, what we decided to do was a more expansive sequence similarity network analysis to see just how many of these trans editing proteins are really, and how ubiquitous are these in the various domains of life. And so we started out with the INS domain of E. coli proline tRNA synthetase and did a sequence similarity network analysis. And what we showed was that there are actually 9 families of homologous domains, putative editing domains. About 72,000 sequences went into this analysis, and we found that Y-back clustered separately from the INS domain, and it has the cysteine editing activity, as I mentioned. ProXP-ALA, which is usually also a trans editing domain homolog of the INS domain with the same activity, hydrolyzing alanine on tRNA pro. And then 5 other clusters of proteins that cluster separately, so they presumably have different activities, possibly even different tRNA substrate specificities, but they're all homologs of the INS domain. And so today what we're going to focus on is the INS superfamily members that are present in trypanosomes. And what we found was that they encode a ProXP-ALA domain that I'll talk about later, and also one of these so-called cluster 7 proteins. At the time we started this work, this cluster of homologous proteins had not been characterized at all. But the T. brucei, Trypanosoma brucei protein, annotated as MCP3, is part of this cluster 7. And so we're interested in trypanosomes because African trypanosomes are devastating human and animal pathogens transmitted by the tsetse flies. And in the case of T. brucei, as an example, this causes African sleeping sickness. And so we were noticing that the only INS domain superfamily member encoded by humans is ProXP-ALA, and they don't encode a cluster 7 protein. So we thought, well, maybe this would be potentially a good drug target. So I also need to tell you or remind you, if you already knew this, that eukaryotic aminoacyl-tRNA synthetases are grouped into what are known as multi-tRNA synthetase complexes. So each of these complexes that are shown on this slide have been characterized from these different species, but and they don't encode all 20 aminoacyl, or they don't include all 20 aminoacyl-tRNA synthetases. They include different combinations of synthetases, which are indicated by the blue domains here, in addition to the red proteins, which are indicate non-aminoacyl-tRNA synthetase proteins that are found in these multi-synthetase complexes and believed to function as scaffold proteins. So what I'm going to start out here by focusing on is the T-brucei complex, which the core complex has been identified to contain only 3 aminoacyl-tRNA synthetases together with these 3 other non-synthetase cellular factors. And I'll kind of highlight this a little bit more on the next slide, where again the core components are shown in the middle. These are the aminoacyl-tRNA synthetases, and then MCP1 stands for multi-synthetase complex protein 1, 2, and 3. And so these are non-synthetase components of the T-brucei MSC. There are 3 other synthetases that are more peripherally associated with this complex, as shown in this 2013 paper. And so again, as I mentioned, well, I may not have mentioned this yet, but the T-brucei complex actually encodes the 2 INS family members that we identified, which I did talk about earlier. So it includes ProXP-ALA, which is actually part of the ProRS synthetase protein, and it is not inserted into the active site, but it is instead found as an N-terminal extension. And so the INS, this INS fold protein is part of the ProXP-ALA family of proteins. In addition, MCP3 is essentially just the INS fold with an appended N-terminal alpha helical extension. And so we wondered, do these INS superfamily members of the Trypanosoma brucei MSC actually serve as editing tRNA editing domains? And then more generally, what is the role of the MSC in their function? So this role of the MSC in all eukaryotic aminoacyl-tRNA synthetases is still a kind of a major open question in the field. And so we thought maybe we could get some insights by studying these putative, editing domains. So I'm going to be showing you a bunch of deacylation assays. And so in these experiments, we use various tricks to mischarge tRNAs with non-cognate amino acids. So what's shown here is alanine mischarged onto tRNA-pro. And we use either tritiated amino acid or in some way label the body, usually the 3' end of the tRNA with P32. And then we monitor the deacylation, add the enzyme of interest, and then monitor the deacylation of the tRNA. And so what I'm going to show you is that indeed in this type of deacylation assay, both the T-brucei proline tRNA synthetase, which has this appended, ProXP-ALA editing domain, and MCP3 are robust ALA tRNA-pro deacylases. And even if we delete the ProRS domain, we see that the ProXP-ALA domain alone has pretty substantial deacylation activity. And so you can see the K-observed values, and these are single turnover deacylation assays. And so they are, trans editing proteins in vitro. But then, what we noticed was that here we're deacylating alanine that's been mischarged onto tRNA proline. But we noticed that MCP3 in particular really robustly deacylated ALA tRNAs no matter what the body of the tRNA was. So here we tested 5 different tRNAs that were all mischarged with alanine, including ALA, tRNA-ALA. And so you can see that activity is really pretty much very similar to the ALA tRNA-pro deacylation activity. And so, of course, this ALA tRNA-ALA activity especially, is undesired, in vivo. And so we wondered, how is this undesired, really promiscuous deacylation activity regulated in cells? And so we kind of went back to look at our multi-synthetase complex, and we thought that maybe MCP3, this promiscuous deacylation activity, is regulated by other members of the complex. And so while we haven't yet tested it in the context of the entire complex, we performed the deacylation experiments in the presence of proline-tRNA synthetase. And here you can see that MCP3 alone deacylates very readily. In the presence of proline-tRNA synthetase, that activity is diminished. And then if we do the assay in the presence of alanine-tRNA synthetase, or the cognate synthetase for ala-tRNA-ala, that really completely abolishes the promiscuous deacylation activity of MCP3. If we use a kind of not unrelated human lysine-tRNA synthetase, we don't see that effect. And so, this led us to propose a new role of the MSC, the multi-synthetase complex, in regulating promiscuous deacylation of correctly charged tRNAs. So we then, I want to say one more thing before I go to the next slide, and that is, so in this assay, we tested the activity of MCP3 in the presence of pro-ARS and ala-ARS. We wondered what happened if we did the same activity assay in the presence of MCP1 and MCP2. What were the role of these other non-synthetase factors in the complex? And so when we did this assay, we see that MCP2 really had no effect on the deacylation activity of MCP3. However, MCP1 appeared to accelerate the deacylation activity. So it seemed to be a really positive factor that, accelerated the deacylation of MCP3 alone. But it turns out that our interpretation of this initial assay was totally incorrect. And rather than accelerating MCP3's activity, it actually has deacylation activity on its own. So when we performed this assay, we saw that it was a very robust ala-tRNA deacylase. And so why is this surprising? Well, this was really surprising because that MCP1 doesn't contain an INS, superfamily domain at all, or in fact, it doesn't encode any kind of aminoacyl-tRNA deacylase domain that has been reported to date. And instead, it's primarily an oligonucleotide-oligosaccharide binding fold. In addition to, it has a GST-like extension and then a lysine-rich kind of linker in between these two. Now, MCP2 has a similar OB fold. And so we tested, again, both of these in deacylation activity. And what we saw was that, as I just told you, MCP1 is a robust ala-tRNA prodeacylase, but MCP2 lacks this activity completely. And so I'll explain why that is, why we think that is in a little while. But first, I want to remind you what OB fold proteins are. And I'm sure you're all really familiar with these. These are OB fold domains, are really very small beta barrel motif proteins or domains. They're very ancient. They're very ubiquitous. There's 17 superfamilies of OB fold proteins. And in terms of nucleic acid binding proteins, there's one superfamily. And within that, there have been identified to be two tRNA-related subfamilies. One is the anticodon binding domain family, and the other is the so-called MIF family, which is exemplified by TRBP111, and also homologous to this EMAP2-like domain. And so I'm going to explain what all of these are in a minute. So let's start with the anticodon binding family. If you're at all familiar with aminoacyl-tRNA synthetases, you know that a subclass of synthetases that includes lysine-tRNA synthetase, aspartic acid-tRNA synthetase, have as their anticodon binding domain an OB fold. So in this case, the beta barrel of lysine of all of these synthetases interacts with the outer face of the beta barrel very intimately with the anticodon binding domain of the tRNA. And what we're looking at here is a co-cryo-EM complex structure of the fully modified human lysine-tRNA, human lysine-tRNA in complex with the lysine-tRNA synthetase homodimer. And we published this complex structure in collaboration with Yang Zhang recently. Okay, so this is the anticodon binding subfamily. Now, what about that MIF family of tRNA binding proteins? Well, this is again represented by TRBP111. This protein is really exclusively an OB fold. It doesn't have any other domains appended to it. And what was shown, again, in kind of a surprising structure that was solved by Zhenwei Zhang's lab about a year ago, is that this beta barrel motif interacts with the CCA end of the tRNA with the cavity of the barrel. So the CCA end of the tRNA, which is where the amino acid is attached, projects into the cavity of this beta barrel motif. And in this structure, there was no amino acid bound to it. And the function of this complex was not clear. But I can just tell you that TRBP111 is similar in structure and its close homologue of the OB fold of MCP1 that I've been telling you about. And so in this paper, they proposed that this motif might function as a tRNA chaperone, but the function was really unknown. So what we wanted to then do was to determine or confirm that indeed it's the OB fold domain of MCP1 that's catalyzing this surprising deacylation activity. And so what's shown here is, again, a schematic of MCP1, which has this, what we now know is a tRNA binding domain, which contains within it the OB fold. It has a lysine-rich helix and then a GST-like domain. And so we did truncations initially from the N-terminus. And again, here's the wild-type activity. If we delete the GST domain, we lose a little bit of activity. If we delete into the lysine-rich linker, we lose more activity. But then if we perform deletions from the other end, from the C-terminus, and we delete the TRBP, the tRNA binding domain, which includes the OB fold, we completely eliminate the deacylation activity. And if we delete a little further into that linker, the same result, really. So basically, this confirms that it is the OB fold domain that has the confers this deacylation activity. So with the structure in hand, we could then look more closely at what are the interactions of the OB fold with the CCA end of the tRNA. And there are three residues that really seem to interact directly with the terminal, 3' residues of the tRNA. And those are highlighted here in the structure. And then the sequence alignment is shown here, where these are, fairly highly conserved residues within these MIF family of proteins. So TRBP is shown at the bottom. Our Trypanosoma brucei MCP1 is shown above that. And then MCP2, which lacks the amino isolation activity, also lacks two of these conserved residues. You can see, and so basically when we mutated, so the next thing we did was mutate these residues in the context of MCP1, and we showed that sure enough, that those mutations eliminate the deacylation activity. So these are critical for binding and activity of this protein. So we also showed, so most of the assays I've showed you so far are deacylating alanine off of different tRNAs. And then we looked a little bit further into the amino acid specificity or preference for MCP1. And what we showed is that it really prefers small amino acids. So in addition to deacylating ALA, tRNA ALA, and ALA tRNA Pro, again, the free enzyme can do both of these. It also deacylated glycine and serine that were mischarged onto tRNA ALA, but it didn't deacylate very well cysteine, 2-aminobutyric acid, or proline. And so it really has a size preference for these smaller amino acids. And glycine and serine are not misactivation errors of proline tRNA synthetase, but they are errors of alanine tRNA synthetase. So we think that these domains could correct both tRNA Pro and tRNA ALA mis-amino isolation errors. And so just to summarize what I've showed you so far is that there are actually now we know 3 different members of the Trypanosomal brucei MSC that have this proofreading activity. So 2 of them were predicted in advance to be editing domains. These are the INS-like belong to the INS-like superfamily member, and that includes the pro-XP ala domain of pro-RS and the INS like protein MCP3. But then surprisingly, we found that MCP1, an OB fold protein, has very similar activity. And so why does T. brucei encode such robust and redundant, ala, gly, ser, tRNA, proofreading proteins? And so we're still trying to understand this, but we have some initial data and some initial ideas on this, I guess. And what I need to tell you now is a little bit about the life cycle of, very interesting life cycle of trypanosomes. So they go through several different stages in their life cycle. So in the insect stage, basically in the tsetse fly, they use proline as a carbon source for synthesis of ATP in the mitochondria, and the byproduct of proline is alanine. And interestingly, in this stage, they also have a very glue pro-rich protein coat, which is important for the protection of fly. In the mammalian form, in the bloodstream form, they use glucose as a carbon source for synthesizing ATP. And here they have a variant surface glycoprotein. And in this intermediate stage, they actually have a very alanine-rich protein coat. And so we wonder whether, perhaps to avoid mistranslation of proline codons and also alanine codons, this deacylation activity might play an important role. And so we first wanted to knock out these proteins in the trypanosomes and see what is the effect on parasite growth. And what we found, and this is work done in collaboration with Juan Alfonso's lab, and what we showed here is that knockout of any one individual protein really had no effect on parasite growth under kind of lab growth, conditions. However, if we then, in addition to knocking out MCP1, knock out MCP3, which is the other trans-editing protein, we now begin to see a growth defect. So this is a TET-inducible knockout system. And so we are seeing a significant growth defect when both of these trans-editing domains are knocked out. And so in current work, we're isolating the glue pro-rich coat protein in this double knockout and wanted to see whether we actually can detect misincorporation of alanine at proline codons by mass spectrometry. Okay, so that's the end of the trypanosome story, and I just want to spend the last few minutes talking a little bit about other eukaryotic systems. So I've told you that this is the multi-synthetase complex of Trypanosoma brucei and encodes this MCP1, which has the unique OB fold domain that is an editing domain. And what we noticed when we looked at all the different complexes is that all of the MSCs characterized to date have at least one of these OB fold homologous proteins. And so in the case of humans, which is a larger complex, it has 9 different aminoacyl-tRNA synthetase activities and also encodes 3 so-called scaffold proteins. One of them is a close homologue of MCP1 and has been termed AIMP1 in the literature. And then in yeast, which has a very tiny, this is the smallest known multi-synthetase complex, it only has 2 synthetase components and 1 scaffold protein that had been termed ARC1P. But again, that scaffold protein has an OB fold homologous to MCP1. And so we wondered, do these other MSC scaffold proteins possess deacylation activity? And so we started out by looking at ARC1P, which again is part of this very tiny MSC. And previously, ARC1P has been shown to enhance the amino acylation activity of both GluRS and medRS in vitro. And shown here is the enhancement of GluRS activity in the presence of ARC1P. But we wondered, does it also deacylate ala-tRNAs? And when we look at the sequence alignment, those 3 highly conserved and important catalytic residues are present in ARC1P. And indeed, we saw that just like MCP1, it has really robust deacylation activity. So ARC1P is also an ala-tRNA pro-deacylase. Okay, sorry. And so what about the human system? Well, humans are very complex. So they have 8 aminoacyl-tRNA synthetases encoding 9 synthetase activities and 3 scaffold proteins. What's shown here is not a it's not a total structure. It's a model of the complex based on cryo-EM and other structural information on different subcomplexes within the complex. And this model was built by Sung-Hoon Kim's lab. And I'm just going to point out where the AIMP1 proteins are. So the blue protein here is AIMP1. This is our OB fold domain, the putative OB fold domain. Well, it is an OB fold domain, putative deacylase. AIMP2 is shown here, interacting closely with lysine-tRNA synthetase and also AIMP1. And then AMP3 is just this little GST-like domain, which kind of holds, is probably a true scaffold protein holding these proteins together. So let's just focus on AIMP1, our OB fold protein. And again, the AlphaFold 3 model is shown here. Here's the OB fold, and then there's a linker and a long alpha helical region. And AMP1 is in the middle here. And again, it has these conserved catalytic residues. And so sure enough, it has the deacylation activity. AIMP3, which was just that little GST domain, lacks that activity. So what is AMP1? I just have a few more slides and to kind of begin to understand, what is AMP1 potentially doing, either in the context of the complex. And when we zoom in on, where it's located, AMP1 is interacting with arginine-tRNA synthetase and AMP2, and then AMP2 interacts with lysine-tRNA synthetase. So what's known is these proposed interactions that have been validated. So I think we can say these are the interactions that happen in the complex. I just told you that AMP1 deacylates ala-tRNAs. And in previous work from the Fang lab and also our lab showed that a role of the N-terminal domain of AMP2 is to keep lysine-tRNA synthetase in a closed conformation that is aminoacylation competent. So that's already been shown. And so what we were wondering is whether AMP1 also modulates the activity of lysine-tRNA synthetase. And so we were kind of inspired, I'll just mention this first, by work of the Jakubowski lab a long time ago, showing that E. coli lysine-tRNA synthetase is really quite promiscuous in vitro, and it misaminoacylates lysine-tRNA with several amino acids. And we recapitulated that in the case of the human enzyme. So it pretty robustly misacylates tRNA lysine with threonine, alanine, and a little bit with arginine and methionine as well. And this is all in vitro data. So we wondered, what is the impact of adding AMP1 and 2 on this activity? So we focused on alanine because we know that the AMP, these OB fold proteins really prefer small amino acids. And what we showed was that, again, in kind of a separate assay, here's the misaminoacylation activity of lysine-tRNA synthetase alone. When we add AIMP2 to this reaction, again, that helps the synthetase become more aminoacylation competent, as was previously shown. And it turns out that it's also more competent in misaminoacylating alanine onto tRNA lysine. But if we add AMP1, it has the opposite effect, and it really prevents misaminoacylation. So this is just a start. We still have a lot more to do in terms of really figuring out what these proteins are doing, both in the context of in the MSC and if they are released from the MSC, which happens under certain cellular conditions. So I just want to end by concluding that we've identified a new class of OB fold tRNA editing domains, that this editing activity is conserved across eukaryotic MSC scaffold proteins, which appear to be much more than just scaffold proteins. And that the new roles of the MSC might be to regulate both misaminoacylation and promiscuous deacylation. And so future questions include, coming back to the trypanosome system, what is the impact of knocking out these proteins on mistranslation in cells? And again, we're looking at the coat protein for starters to see if that is, has a lot more mistranslation happening. And then do these synthetases and editing domains possess the same activities when they're MSC bound versus free form? So we're trying to isolate these in the context of the full MSC, and we're working on that now. So I just want to thank again the people, the 3 main contributors in my lab to this work, and then Gabriel in Juan Alfonso's lab for some of the trypanosomal brucei in vivo studies. just highlighting here some of the views of our campus here at Ohio State University, and shown over here is the kind of central part of campus, the oval, but we all know, of course, that the central part of campus is really on Saturdays at Ohio Stadium. So, so with that, I'll end. I'm happy to take any questions. Thank you.
Thank you, Dr. Musier Forsyth, for the insightful presentation. So we now have about 10 minutes for questions. I'd like to remind everyone that you can submit your question using the Q&A feature if you haven't done so. So let's see. We have one question in the Q&A. Is there a reason MCP1 was deleted completely in the trypanosome life cycle experiment instead of simply mutating the MCP1 active site residues for deacylation?
Well, we could certainly do that. In the context, it's actually not that difficult to do complete knockouts in the trypanosome system. And again, Juan Alfonso's lab did the CRISPR knockouts of those. We could go in and specifically mutate specific residues as well. But we thought, for starters, we would just look at the complete knockout to see the most hopefully dramatic effect. And it was kind of interesting that there was no phenotype until we knocked out a second redundant editing domain. And so we're now also doing those types of in vivo studies with kind of looking at the potential an imbalance of amino acids. So what happens when we really do have a lot more alanine over proline in the system? And again, we want to see if there is going to be mistranslation in cells under those conditions. So I guess to answer your question, I don't think that if we didn't see a phenotype with a complete knockout, we probably wouldn't see one with with just the catalytic domains mutated. So
okay. Thank you, Dr. Musier Forsyth. Maybe I have a question. So maybe more generally, like so understanding the role of the OB fold containing proteins, like could it lead to potential like biomedical or therapeutic applications?
yeah, I think so. I mean, all of these it seems like all of the eukaryotic enzymes encode these. So I think if you could kind of figure out what's different about them. I actually still think MCP3, which is kind of a unique editing domain in the context of trypanosomes that is not encoded by humans, is maybe the better therapeutic target. But yeah, I think potentially we could do that. Now we still have to do a complete, I think, more careful bioinformatics analysis of all of the OB fold, MIF family members to see how widespread this editing activity, really is beyond the components of the MSC. So that's something that we're going to be working on. But yes, potentially, I think.
Thank you, Dr. Musier Forsyth. We got another question from the Q&A session. It's human AIMP1 appears to have a long disordered region in its AF structure. Does it form condensates in human cells?
Oh, that's a very good question. I am not sure about that. I don't think I have read anything about that. So, but that's a very good question. And I think, it has been shown that when cells are stimulated in different ways, that these AIMP1, almost all the synthetases in the MSC can be released. And under those circumstances, they could have activities, and they have been shown to have activities that are not related to tRNA charging at all. And they could have, different, and they have different moonlighting functions. And so I think that's certainly possible. Here I'm showing again the MSC. So yeah, they're very long. They're helical, but there's some disorder, especially in the linkers as well. So I think that's certainly possible. And right now I'm not recalling that I've read anything about that specifically. But again, they can be released, and I think that under those conditions, I think they could form condensates potentially. It's a good idea.
Okay. Thank you, Dr. Musier Forsyth. I think that's all the questions we have. So thank you for everyone to submitting the solvable questions, and Dr. Musier Forsyth, thank you for your detailed responses. And so Dr. Musier Forsyth, I would like to hand it back to you for any final thoughts for key takeaways before we conclude today's session.
Okay, well, I just want to thank everybody for joining today. And if you have any other questions that you didn't have a chance to ask in the Q&A, feel free to just email me. You should be able to easily find my email address, and hopefully we'll have a lot more insights into the role of these MSCs and these trans-editing domains in the future. So thank you again.
Okay. Thank you so much, Dr. Musier Forsyth, for your time expertise today, and for all of you joining us. Be sure to keep an eye out for the upcoming webinars in our Breakthrough webinar series. And we hope to see you again soon in November. And have a great day. Bye.